needs. So, the three major challenges we're facing, we humans now time, that are the food, energy, and the environmental degradation. So, the three and all these three are the plant related. All of our food is produced by plants. Plants are the source of energy production and they are intimately involved in climate change and a major factor in a variety of environmental concerns, including agricultural expansion and impact on habitat destruction. Climate change place additional stress on the food supply and on various habitats. So, plant research is instrumental in addressing all of these problems and moving into future. If we are to make headway in understanding how these essential organisms functions and build the foundation of for the sustainable future, then we need to apply the most advanced technologies available to the study of plant life. Plant science has not often been the driver of innovation, but often enough has profited from developments made in other areas. Mm, it gives me a huge pride and pleasure to welcome Honorable Dr. Mehta sir and Dr. Wadikar sir. Sir, welcome sir. Now I request our respected principal sir to give us valuable guidance.
Good morning, all dear participants present on this digital platform. Today, our college, MSP Mandal's Yashwantrao Chauhan College Department of Chemistry has organized Department of Botany has organized one day national level seminar on recent trends in plant science. I cordially welcome all guests, my friends, keynote speakers, Professor Dr. Suryakant Mehta, Professor and Head, Department of Botany, Mizoram University, Mizoram, other resource person, my colleague and friends, Dr. Madhukar Wadikar, Professor and Head, Department of Botany, R.B. Attal College, Georai, Bid, Maharashtra. My colleague, Vice Principal Dr. Ramesh Shinde, Professor P.K. Jadhav, IQAC Coordinator Dr. Mukund Rajpankhe, convener of this seminar, Ms. Gayatri Kadam, technical supporter Mr. Ali and Asif Sheikh, and all online joint delegates in this academic events. Our college has established 1972 by Marathwada Shikshan Prasarak Mandals to fulfill the requirement and hunger of knowledge of stakeholders of Ambazogai and around villages. I heartily thanks President Honorable MLA Sri Prakash Dada and all and Narendra Ji Kale and other CDC member. They all always inspired us to take this type activities. Apart from our college has a research center in English, political science, Marathi, Hindi, and commerce. 17 research guides and 72 researchers are contribute to the research process through these three research centers. Apart from this year, we have organized other research centers. No doubt, from the time of establishment till this date, the graph of enrollment students in increasing order. Our college has awarded A+ grade with CGPA 3.45 in fourth cycle by National Assessment and Accreditation Council, Bangalore. Recent trend in plant science simply refers to biotechnological development in plants. Best example of this is genetically modified crops, which are produced by modifying plants genome by physical methods using some technologies. Genetically modify crops increase crop yields, need less pesticides, enhance nutrients compositions and food quality, resistance to disease. But constant use of genetically modified crops ultimately cause negative impact on health and ecosystem also. Nowadays, we can observe many people are allergic with regular things. This is a disadvantage of these crops. Genetic modification, tissue culture, crafting are the same technologies with which plant transform for the better yield with less efforts. Tissue culture is production of a new plants from small amounts of plants tissues under controlled laboratory conditions. These techniques produce disease resistance in high yielding varieties of plants. Plant science is a way to fulfill increasing demand for food and energy. Due to this, the food grains imported from outside countries will definitely to be stopped. I hope that we will come to positive conclusion from the brainstorming of this seminar. I think today seminar will be fruitful to all researchers and participants. Knowledge received from eminent and experts in the aspect during the events will be definitely useful for updating of faculties, researchers, and to enhance to the knowledge of students. And once again, best wishes to all participants. Thank you. Jai Hind, Jai Bharat.
Thank you so much, sir. It's really been an honor for our college to organize such a beautiful program with such amazing guests, Dr. Mehta sir and Dr. Wadikar sir. Words are too small to describe their achievements. Now, we are feeling so fortunate to have you both, sir. I request Shinde ma'am to introduce our guests. Shinde ma'am.
Thank you so much, Gayatri ma'am. Good morning, everyone. I am Pranali Shinde from Department of Mathematics. I am here to introduce keynote address and our resource person, Professor Dr. Mehta sir. Professor Dr. Mehta sir has completed PhD in adsorption and uptake of copper and nickel by Chlorella vulgaris. He has a specialization in biochemistry, physiology, algal biotechnology, and microbial biodiversity. Sir worked as CSIR Research Associate, Senior Lecturer, Associate Professor, and now he is working as Professor at Botany Department in Mizoram University, Aizawl from 2012. Sir is the Chairman of Board of Studies of many subjects, NAC Committee in Mizoram University. He is awarded JRF, SRF, UGC, Research Associateship, and also Postdoctoral Fellow, Nanjing Agricultural University, Nanjing, China. He is also a research guide. 5 students awarded with PhD under his guidance, and 8 students are working for doctoral degree. He has a 23 years of research experience. Dr. Mehta sir completed almost 8 research projects and 24 research paper were published. It gives me a huge pride and pleasure to introduce our guest of honors, Dr. Mehta sir. Thank you.
Thank you, ma'am. To know more about recent trends in plant science, we would like to call respected Mehta sir to deliver speech on the topic. Mehta sir. Mehta sir.
मेहता सर। मेहता सर, आवाज आ रही है आपको? मेहता सर, आपका माइक ऑन है, बात कीजिए। मेहता सर, आपका माइक ऑन है, बात कीजिए।
Video phenomena आहे त्याचं. नहीं सर, आवाज अभी भी आई क्या?
हेलो हेलो
हाँ मेहता सर आपकी आवाज आ रही है सर।
आ रही है ओके
जी जी जी।
ओके सर ओके जस्ट 1 minute so now is fine sir audio and video both hello
जी सर।
uh, this okay na sir
यस यस।
okay okay hello
हाँ सर।
so we will start
बिल्कुल सर बिल्कुल।
okay so very good morning I am Professor S.K. Mehta and I am very thankful to Principal Dr. Sri Sat, chairperson of this webinar, and I am also thankful to organizer of this webinar, Ms. Gayatri Kadam, Department of Botany. And as introduction of myself, I am Professor S.K. Mehta, and I am working at Department of Botany in Mizoram University. So today I was requested to give a webinar on topic recent trends in plant science. So I welcome all of you and very good morning. Now I am going to start today discussion. So for internet speed, I am going to switch off my video and I will share my presentation. Is I am audible?
जी सर बिल्कुल बिल्कुल।
Yes, sir. Yes, sir.
Okay, okay. So screen, can you see? Hello.
Yes, sir.
Full screen, madam, can you see?
Yes, sir. Yes, sir. We can see.
Just one more check. Please see it is moving or not. Slide is
Yes,
moving.
yes. Yes, side is moving, sir.
Okay, thank you.
Yes.
So today talk is about recent trends in plant science. I try to give my presentation over these points. First, I will highlight overview of recent advances. Then there are following topics like molecular insight into plant physiology. I will highlight what are recent developments, what is direction of research. Likewise, innovations in crop improvement. We will discuss the recent advancement. Then one important aspect is plant microbe interactions. So I will highlight that aspect also. And advances in genetic engineering. Then environmental adaptation in plants. What types of research are going on and what we should further do in area of environmental adaptations in plant. And I will summarize under heading future directions in plant biology. So let us start. First of all, that why plant biology matters to us. So most important thing is that plants most important thing is that plant are oxygen producing organism, autotrophic organism. They produce oxygen, and that oxygen is the source for other organisms, including human. Through the photosynthesis, plant absorb carbon dioxide, and by this way, they help to mitigate climate change. Next is that plant are helpful for food supply to us as a population expand. Ensuring stable, nutritious food supply is very imperative. So, understanding plant genetics, physiology, and interaction with environment help in creating crop adapted to pest, disease, and environmental stressors. So, innovations in crop improvement guided by plant biology research contribute to global food security. And plant are very important in environmental balance. Plant play very important role in maintaining environmental balance and ecological stability. So, where is plant biology? Plant biology is at the intersections of global challenges and their solutions. So, there are global challenges like population growth, food security, and climate change. And plant help to mitigate or plant provide a solution in form of environmental conservation, sustainable agriculture practice, and resilient crops. So plant are at intersection where there are global challenges and where solutions are required. As we navigate the complexity of 21st century, understanding and advancing plant biology is not just a scientific endeavor. It is a fundamental step towards ensuring a balanced, sustainable, and nourished world for generations to come. Plant science, as we will see in coming slides, is a dynamic and ever evolving field that continually adapts to new challenges and embraces emerging opportunities. So, there is continuous research and development in the field of plant biology. This dynamism is propelled by a combination of technological advancement and interdisciplinary collaborations, which together have fueled rapid progress in recent years. So, there are three major technological advancements which have contributed much in development of plant biology. First is genomic tools. Through these tools, for example, CRISPR-Cas9 or high uh, throughput sequencing, now we are able to modify plant genome at very high precision. We can alter genome sequence by single nucleotide. So genomic tools have added much in progress of plant biology. Second technological advancement is imaging technology. Now we have good imaging technology, for example, confocal microscope and living cell imaging. By this imaging technology, we are able to monitor live changes in cell physiology and morphology. Third advancement in technological aspect is omics approach like proteomics, transcriptomics, and metabolomics. Advancement in these omics approach have enabled plant biologists to evolve to discover new aspects of plant adaptations to different environmental conditions, to develop high yielding crop varieties, etc. So there is technological advancement during recent years which have helped much in development of plant biology. Before going in detail about what are recent development, just here I want to highlight milestones that have shaped our understanding of plants and their pivotal role. So first, if we see that long back BC, Theophrastus, he wrote Inquiry into Plants and on the causes of plants that laid foundation of the scientific study of plants. Now, during 1630 and 1650, microscope was invented, and microscopic observation by Robert Hooke and Marcello revealed that cellular structure of plant. Further development came in the form of a book Species Plantarum written by Carl Linnaeus, where he introduced a proper nomenclature of plants. After that, if we look during 1827, Robert Brown discovered cell nucleus. That was a milestone in plant biology. Then we are moving forward. Then all of we are known about Charles Darwin's contribution in the form of a book on the origin of species that laid the groundwork for evolutionary biology, plant evolution, and adaptation. And further, when we are moving to 90s, then there was work by botanists like Hugo de Vries, Carl Correns. They worked on a mutation and they rediscovered Gregor Mendel's law of inheritance, and that established the basis of modern genetics. Then moving to 1930s and 1940s, then there was discovery of techniques like X-ray crystallography that lead to study of in detail study of biomolecules like chlorophyll, structure of protein, etc. And after that, during 1950s, 60s, there was a green revolution for high yielding crop varieties. Further moving ahead, during 1980s, that decades, there was much work on genetic engineering techniques. So manipulation of DNA, then development of recombinant DNA technology, then uh, development of genetically modified plants that happens during 1980s. And after that, further development was that in 1996, first GMO crop, soybean crop, that was GMO, was released, making a milestone in biotechnology impact on agriculture. During 2000, that first decade, there was a completion of Arabidopsis thaliana genome project, and that laid the foundation for plant genetics and molecular biology, serving as a model organism for plant research. Further, and most recent development in plant biology is during 2010s onward, invention of this CRISPR-Cas9 gene editing technology. That technology has revolutionized whole plant biology. By using this tool, now scientists are able to edit gene for different purposes. So these are some background for gradual but significant development in plant science. As in first slide, I highlight that we will discuss these developments in a few topics. So first topic is that what is recent trends in molecular insight into plant physiology. So we will discuss with a few slides. First is that genetic regulation. Then is epigenetics, and third is plant hormone. So here under genetic regulation, there is much development due to discovery of CRISPR-Cas technology. The revolutionary CRISPR-Cas gene editing technology has allowed precise manipulation of plant genome. Researchers can now target and modify a specific gene with unprecedented accuracy, enabling the development of crops with enhanced traits such as disease resistance and improved yield. So this technology, CRISPR-Cas9 technology, is a revolutionary discovery in the area of genetics and molecular biology. Now, what is this? In very briefly, with one slide only, I will highlight that what is this technology, because this tool is very powerful. With this tool, we can target a particular gene, a particular nucleotide in DNA, and we can modify. So, what is this tool? This tool is simple, and this CRISPR-Cas9, this one is a Caesar-like tool that already exists in bacterial genome. Actually, bacteria have these tools to defend from attack of the viruses. So that benefit now we are using for genetic engineering. So this Cas9, this one is enzyme or a protein, and this cut DNA double strand at a specific place. Now question is that how this Cas9 will identify this place only, this will cut only here, not here, okay? This is assisted by a guided RNA. So this RNA is very complementary to target sequence, and this RNA is attached to this enzyme Cas9 protein, and this guide Cas9 to attach at this place. And after attaching here, this Cas9 cut a double strand. Now DNA is fragmented by cutting off double strand. This cutting is very precise, very accurate, and it cut only where we want to cut. And This cut end can be joined naturally by cellular mechanism. At this point, now we are able to insert new programmed DNA fragment here. This may have some gene for environmental adaptation, for pest resistance, for fighting against pathogen. So this technique is simple, very cost effective, and it requires very short time to modify the gene of organism. So this has revolutionized our genetic engineering and plant biology. Under genetic regulation, further development and research is in area of functional genomics. So functional genomics is a discipline where we can modulate, we can modify the expression of a particular gene. So advances in functional genomics, including high throughput sequencing and genome-wide association studies, have provided comprehensive insight into the function of a specific gene. This has accelerated the understanding of complex genetic network controlling plant development, stress response, and metabolism. Through functional genomics, we are now able to knock down certain genes which inhibit plant productivity. Likewise, there are many area of research where functional genomics is helpful in improvement of crop varieties. This high throughput sequencing methods, there is great development in this area. So we have now different sequencing approaches. A recent one is that new generation sequencing, new generation sequencing, and RNA-seq technology. So before, with traditional sequencing techniques, it was possible only to sequence a small fragment of DNA or a small portion of gene. Now, due to advancement in technology, sequencing technologies, now it is possible to sequence a long stretch of genes or genome. So RNA-seq technology has also revolutionized our functional genomic approach. So what is the implications of genetic regulation development? So tailored crop traits, the ability to edit a specific gene allow the creation of crops with desired traits such as resistance to pest, disease, and environment. So using CRISPR Cas9 tools, now we are able to edit specific genes, and that editing is for a particular objective, like we want to create a crop with resistance to pest, resistance to disease, or environmental stress. And moreover, this advancement in genetic regulations have accelerated breeding program. For evolving development of one crop by traditional breeding take years of research. But now, due to advancement in genetic regulation techniques, now it is possible to get objective of this breeding in very short time. So CRISPR Cas technology has expedited traditional breeding program by reducing time required to develop crops with improved characteristics. There are a lot of research in this area for CRISPR-Cas9 based genome editing. This has a revolutionized approach for crop improvement and global food. So this review cover a very wide, vast area focused on CRISPR-Cas9 technology. So there are certain important crop traits like disease resistance, yield, stress tolerance are being enhanced in various species such as Triticum aestivum, Oryza sativa, Zea mays, and Solanum tuberosum. So these new tools are tested for the development of different types of crops for disease resistance and increased yield. Second, advancement in plant physiology, molecular plant physiology is development of epigenetics approach. So what is this epigenetic approach? The word epigenetic literally means that in addition to change in genetic sequence, as I said that uh, improved crop can be developed by changing sequence of gene. That is one approach. Alternative to that approach is epigenetic approach where there is no change in sequence of gene. For example, every plant has all genes, but flowering in a plant is at a particular stage of life cycle of plant only. Why it happens? Gene is already in plant, so why flowering is set at a particular time? This is due to epigenetic control of the gene expression. Gene is present there, but that not express due to certain mechanisms. So its expression is halted for some time. Same natural approach is now being used for improvement of our crop varieties. So epigenetic modification is modification in expression of gene, targeted gene, without altering gene sequence. So the term has evolved to include any process that alter gene activity without changing the DNA sequence and lead to modifications that can be transmitted to daughter cell. It is not well established that epigenetic modifications are transmitted to daughter cells. There are certain proofs that epigenetic modification can be reversed back also. So this epigenetic approach has contributed much in plant biology. The best known epigenetic process is DNA methylation. It is well established mechanism of epigenetic process, and this is the addition or removal of methyl group predominantly where cytosine base occur consecutively. Another significant epigenetic process is chromatin modification. So research are going on in area of epigenetics by either methylation of DNA or by chromatin modification. Objective is to evolve crop that may be resistant to pathogen, may be resistant to pest, and adapted to environmental conditions. In epigenetics, nowadays there is also huge research on application of microRNA and small interfering RNAs, siRNAs, to modify plants for stress resistant plant development. So these two application of microRNA and small interfering RNA, they modify the gene expression, but there is no change in gene sequence. So it is epigenetic approach. This modification alter chromatin structure to influence gene expression. Likewise, there are a lot of work in epigenetics, and you can find several papers that describe the epigenetic regulation in plant for stress response in non-model plant as well as model plants like Arabidopsis. So in recent years, enormous progress has been made in understanding the role of epigenetic regulation response to environmental stimuli, especially in response to stress. Modification of DNA methylation profile in plant can cause phenotypic variation. This is another important area of recent research. Now, DNA methylation profile is in natural in plant, but when we modify natural DNA methylation profile, that alter phenotypic variation. Stress can also result in change in DNA methylation. So there is a research in this area to work out on how different stress condition changes DNA methylation. DNA methylation content can also be regulated in response to abiotic stress. So experiments in maize and Arabidopsis shows that cold stress might induce modification of DNA methylation status. Likewise, there are many research work focused on effect of different kind of biotic and abiotic stresses on DNA methylation status of different plant species. We are known about process vernalization, some type of cold treatment, so that induce flowering. Actually, behind vernalization treatment is the modulation of or reduction of level of DNA methylation and induce the initiation of flowering. So instead of vernalization, now we can modify using certain tools the DNA methylation in plant, and that modification may result into enhanced flowering in plant. So in this area, a lot of works are going on in area of epigenetics. Third, advancement in molecular plant physiology is about plant hormones. So understanding of hormone signaling network, this is another important area of research during recent years. A lot of work has been done and are being done in area of hormone signaling networks in response to different stress condition, in response to nutrient availability in soil, etc. So there are many good research paper and a review paper that very thoroughly describe that what are recent advances and emerging trends in plant hormone signaling. Advances in molecular biology and omics technology like proteomics, transcriptomics have provided a comprehensive understanding of hormone signaling network. Researchers can now elucidate how different hormones interact and modulate complex physiological process in plant. So these are another area of research nowadays that botanist, molecular biologist, they are trying to understand the network of hormone signaling in plant in response to different type of stresses and physiological conditions. Further, there is approach that called synthetic biology applications. So researchers are engineering plants to respond to synthetic hormones, enabling precise control over growth, development, and stress response. So also work is going on in area of synthetic hormones. So there is increasing research to understand signaling crosstalk between biotic and abiotic stress responses. So there are different signaling pathway, signaling molecules like ethylene, abscisic acid, salicylic acid, jasmonic acid. These pathway are induced by some kind of stress, but some pathway they interact, they communicate to each other. So overall there is a high complex network of hormonal signal transduction. So researchers throughout the world are now studying to understand this signaling crosstalk, and this may be helpful in development of stress resistant, pest resistant, and high yielding crop variety. So there is much work going on understanding of hormone signaling networks in different plants in response to different stresses. Today, synthetic chemicals, including plant growth regulators, are used to study and manipulate biological systems. So, this is another area of research in plant biology where synthetic chemicals are used to study and manipulate biological system. So this collectively is called as chemical biology. So you can find many papers that are around this new area of plant biology. So integration of these breakthrough in genetic regulation, epigenetics, and plant hormone is paving the way for personalized agriculture, where crops are tailored to specific environmental conditions and desired traits. Ongoing research is focusing on utilizing these insights to address global challenges such as climate change, food security, by developing crops that can thrive in diverse and changing conditions. So, second area is innovation in crop improvement. Now, recent years, a lot of work is going on for crop improvement. So, cutting edge technologies are revolutionizing crop improvement, entering in an era where precision, speed, and innovation converge to address challenge of growing global population and environmental constraints. From manipulating the plant genome to harnessing data-driven insight, these technology are reshaping the landscape of agriculture in profound ways. So, what are innovation in crop improvement is that precision breeding techniques work is going on about this, high throughput sequencing, phenomics and remote sensing, gene editing for pathogen resistance, machine learning and artificial intelligence, advanced breeding platform. So these technology are now being developed and evaluated for crop improvement in different crops. So, what is precision breeding techniques? So, advanced genetic tools such as CRISPR-Cas9 enable precise and targeted modification within the plant genome. This level of precision accelerates the development of crop with specific desirable traits, ranging from enhanced nutritional content to resistant against pests and diseases. And I have discussed about high throughput sequencing. So that development has resulted into identifying key genes associated with desirable traits. Next is phenomics and remote sensing. So phenomics and remote sensing technology allow for comprehensive analysis of plant characteristics from molecular level to field scale. This data-intensive approach enables researchers to monitor crop performance in real time. So a lot of work is going on in monitoring crop performance in real time using remote sensing technology. Next is gene editing for pathogen resistance. So here we have good tools to edit gene to modify specific genes related to disease susceptibility. Machine learning and artificial intelligence, these are transforming data analysis in agriculture by processing vast data set related to climate, soil, and crop performance. This technology provide predictive insight, helping farmers make informed decision on planting, irrigation, pest control, ultimately optimizing crop yield. So this is new area of research where how the machine learning and artificial intelligence can help in development of uh, improved varieties. Advanced breeding platform. So here there are different breeding platforms. These platforms may involve combination of genetic data, robotic automation, advanced analytics to rapidly and efficiently select and propagate superior plant varieties. Next area of uh, recent research is in-plant microbe interactions. Plant microbe interactions play a crucial role in shaping the health and productivity of plants. So recent trends in this field have witnessed significant advancement with a focus on understanding the complexity of these interactions at molecular, genetic, and ecological levels. So now research is going on to understand plant and microbe interaction at the level of molecular, genetic, and ecological. So under plant microbe interaction, there are different area of research. So one is that to research about microbiome. So many plant biologists, microbiologists, they are working on this aspect. So holobiont concept, here the emphasis on viewing plant as a holobiont consisting of plant host and its associated microbial organisms. So understanding the dynamics of plant microbiome and its influence on plant health, development, and stress tolerance is a growing area of research. Second, under plant microbe interaction studies, there is approach called multi-omics approach. So here there is an integration of metagenomics, metatranscriptomics, and metabolomics to comprehensively study the plant-associated microbial communities. This study allows researchers to identify specific microbial function and their impact on plant physiology. Another area of research going on under plant microbe interaction is to study symbiotic relationship. So one example is arbuscular mycorrhizal fungi. So there are in-depth studies on the role of arbuscular mycorrhizal fungi enhancing nutrient uptake, particularly phosphorus. So understanding the signaling mechanism between plant and AMF is another area of research. And what is impact of this AMF association on plant growth and stress response? So many uh, plant botanists, many molecular biologists, microbiologists, all they are working in this area. And another is nitrogen fixing symbiosis. So exploration of nitrogen fixing symbiotic interactions such as those with rhizobia and actinomycetes is another area of research. So strategy is to enhance nitrogen fixation in crop and reduce the need for synthetic fertilizers are gaining attention. Then is under plant microbe interaction, there is another new area which is fall under plant immunity and defense. So pattern recognition receptor, many work is going on this pattern recognition receptor. So there is investigation of the plant recognition microbial pattern and activation of immune response. So discovering the intricate signaling pathway involved in plant immunity against pathogen. This is a new area of research. Microbial effector and plant response. So understanding how pathogens deploy effectors to manipulate plant defense and how plant counteract these strategies are now focused. Insight into co-evolutionary arm race between plant and microbes. Much development has been done in the area of biological control agent. So, development of biopesticide and biofertilizer to promote sustainable and eco-friendly agriculture practice. Uh, Next is microbial induced systematic resistance. This is another area of research during recent years. So investigating the mechanism through which certain beneficial microbes prime the plant immune system. Means that when plant are exposed with very low dose of microbes, so plant remember that interaction, and in later phase of life cycle, if there is a severe attack of that microbes, plant remember that response like our immune system and resist that microbes. So this priming micro priming pattern That is a new area of the research. And plant microbe interaction, rhizospheric interaction. So many workers, researchers are focusing on the understanding of the chemical signaling and communication between plant and microbes in a rhizosphere. Another aspect in plant microbe interaction is to study that what is effect of plant microbe interaction in soil health and fertility of the soil. So strategy to harness these interactions for sustainable soil management and carbon sequestration. So many work in going on in chemical signaling and impact on soil health. So as research in plant microbe interaction progress, the integration of these findings holds promise for developing innovative strategies for sustainable agriculture, reducing reliance on agrochemicals, and enhancing crop resilience in face of environmental challenge. So work has to be done in this area. Exploration of these trends is essential for unlocking the full potential of plant microbe interaction in shaping the future of agriculture. Now coming to advances in genetic engineering. So during last 2 decades, a lot of work has been done in genetic engineering. So advances in genetic engineering have transformed the field of biology, enabling precise manipulation of genetic material of organism for various purposes. In the context of plant, these advances have significant implications for agriculture, medicine, environmental sustainability. So with 3-4 slide here, I am giving some key recent advances. One I have discussed about CRISPR-Cas9 technology. This has revolutionized this genetic engineering. Then is multiplexing. So with traditional techniques, we can modify one gene at a one time. But this CRISPR-Cas9 technology, this can target multiple genes simultaneously. This is important because many stress response and crop productivity, pest resistance are multigenic trait. So only modifying one or some selected genes does not fulfill objective. So it is required to manipulate uh, several genes, related genes for stress resistance likewise. So there is multiplexing approach where several genes, multiple genes simultaneously can be modified. So there is a great development in this area, and this is very recent development. And there is a great development in epigenomic editing. So beyond DNA sequence change, CRISPR technology is increasingly applied for epigenome editing. So a researcher can modulate gene expression without altering underlying DNA sequence. You see, this one is important in the sense that there are a lot of issues related to GMO, genetically modified organism, where foreign DNA is inserted. So gene sequence is modified. So there are ethical issues. But here in epigenomic editing using CRISPR technology, there is no insertion of foreign DNA into the plant. So it looks safer than GMO. So a lot of focus is on this CRISPR technology for genome editing or gene editing. So under genetic engineering, there is much advancement in this pest management. So here, gene drive technology has the potential to alter or suppress population of pests, not the single one or two species. So gene drive technology, now work is going on around this gene drive technology to manage several whole pest population. This can be harnessed for pest control and disease prevention in crops. Further advancement in genetic engineering beyond the genetic editing is now that synthetic biology techniques are employed to design and construct custom genetic circuits. This allows the creation of a plant with novel function such as enhanced stress resistance and improved nutrient utilization. So much work in going on this new approach where new genetic circuits are constructed for several purposes. Another is biofoundries. So automated platforms known as biofoundries facilitate the high throughput construction and testing of genetic construct. Otherwise, before years are required to construct modified plant, genetically modified plant, and testing of that plant for stress tolerance and anything. But due to this new development biofoundries concept, now this can be done in a very short time. So genetic manipulation without altering gene sequence, without inserting foreign DNA, and next testing of this new modified crop can be done in very short time using biofoundries concept. And another advancement in genetic engineering is RNA interference, either by RNAi technology or small inhibitory RNA technology, siRNA. So RNA technology is utilized to target gene silencing, enabling the down regulation of specific gene. So a lot of work has been done and continuous work is still continuing on RNA technology, application of RNA technology for evolving high yield crops, evolving crops that fruit that can be transported for long distance. This RNA technology is very useful. And functional genomics, RNAi is extensively used in functional genomic studies. Researchers now can do gene knockdown, means one gene expression can be inhibited. So like best example is tomato. Nowadays tomato can last for 15 days, 20 days. It not get soft because there is one gene for pectin that is knocked down, so fruit not get soft. So there is a lot of development in gene knockdown technology for improved crop varieties. Further advancement is creating entire genome. So advances in genome synthesis enable creation of entire plant genome from the scratch. This technology has implication for designing plants with custom genome optimized for a specific function. And chloroplast transformation. Much work has been done and is still continuing on genetic engineering of chloroplast that allow the introduction of foreign gene directly into plant plastic DNA. And trans stacking, this is new concept where plastic engineering facilitates the stacking or storing of multiple transgenes within the chloroplast. So enabling the incorporation of multiple traits in a single transformation event. So much work is now in this area. Above all, there is a much advancement in genetic engineering, but always there are ethical issues in genetic engineering. So we are not going to discuss this aspect. Future direction in genetic engineering. So the future of genetic engineering in plant holds promise for addressing global challenges such as climate change, food security, sustainable agriculture. Continued research will focus on refining existing technologies, addressing ethical considerations, and exploring new avenues for manipulating plant genomes to meet the evolving need of society. Briefly, we will describe advancement in environmental adaptation in plant due to climate change. So, recent trends in understanding environmental adaptation in plant have been marked by interdisciplinary approaches, advanced technologies, and a focus on addressing the challenge posed by changing climate. So, there are different stresses exposed by climate change are natural, like drought, salinity, nutrient. They have when plant respond to these stresses, and these stresses reduce growth. In response to these stresses, plant induce molecular chaperones to defend these stresses, and there is metabolic adaptations. So all are these area of research where scientists are working that how drought stress will affect stomatal closure, how different environmental stresses affect antioxidant production, and how plant adapt. So there are great work on these aspects to understanding the impact of environmental stresses in plant, and that will help in development of crop varieties that that may tolerate different types of stresses. So researchers are employing genomics tool to identify genes associated with climate resilience in plant. So many laboratories are working on this aspect just to identify which genes are associated for climate resilience. Then transcriptomics and metabolomics, the high throughput techniques like RNA sequencing, metabolomic profiling are used to honor complex molecular response of plant to environmental stress. So this is another niche of research work. So many reviews paper are available where we can see that how understanding plant stress memory response for abiotic stress resilience. So great development is done in this area. Microbial partnership investigate another area is investigating the role of plant microbe interaction in environmental adaptation. So understanding how beneficial microbes in rhizosphere contribute to nutrient acquisition, stress tolerance, and overall plant growth is a focus of the plant biology research. Synthetic biology application. So researchers are now exploring synthetic biology approach to engineer plant with enhanced ability to form beneficial symbiotic relationships. So you see papers, review papers are available which have compiled development in plant biology in the area of microbiome engineering. many reviews papers are available. So environmental adaptation, epigenomics research is also helping in understanding of the environmental adaptations in plant, and as a result creating new technology for more adapted plant species. So here is just a epigenetic process in plant stress priming, open question and new approach. This review paper have summarized that how epigenetic approach can be used for evolving plant varieties that may be able to tolerate climate change. There is another research in area of digital agriculture and precision farming. So modern tools are being used. So there is sensor technologies. So advances in sensor technology are utilizing for real-time monitoring of environmental conditions. This include monitoring of soil moisture, development of sensor for satellite imaging. So all these are integrated for this digital agriculture. So digital agriculture is a new area of study, research, data analytics and machine learning. Now the integration of big data analytics and machine learning algorithm help analyze large data set from environmental sensor. Designer plants. So researchers are exploring the design of plant with synthetic genetic circuits that confer specific environmental adaptations. This include engineering plants to respond to specific stressors or optimizing metabolic pathway for enhanced resilience. And biohybrid system. So integrating synthetic biology approaches with natural plant process to create biohybrid system. This involve incorporating engineered microbial partner or synthetic signaling pathway to enhance the plant adaptation. So now work is going in this area also to develop designer plant and in a biohybrid system. So some papers are available where you can read in detail about concept of designer plants for climate resilient and for other purpose like phytoremediation. Then is another area of research in environmental adaptation is assisted migration. So, studying the feasibility and implication of assisted migration, where plant population are relocated to area with more suitable climate, means introduction of crop variety from one environment to other environment. This is another area of research and it is called assisted migration. So this is explored as a strategy for conserving plant species threatened by climate change. And another is ecological restoration. So research focus on developing strategy for restoring ecosystem impacted by environmental changes. So a lot of work is going on in this area also. So here you see in this one, this is example of ecological restoration. A mangrove vegetation, see 2008, nothing was there. It was degraded, whole ecosystem. But due to restoration approach, now vegetation started to growing. So this type of work is also going on throughout the world about ecological restoration. So in area of environmental adaptation, Recent trends in environmental adaptation in plant are moving toward a holistic understanding of plant response to dynamic environmental conditions. The integration of advanced molecular techniques, synthetic biology application, and data-driven approaches positions plant science at forefront of addressing the challenges posed by climate change and ensuring global food security. So overall, it is a summary of presentation that what is the future direction in plant biology. So just with two slides, I have 5, 6 points. First is future direction is to understand the genetic basis of climate resilience in plant and developing crop with enhanced tolerance to various stresses. Second approach maybe like utilizing synthetic biology to engineer plants with custom traits for specific climates. And third is that research should be done on the use of sensor, drone, and satellite imaging for real-time monitoring of crops, enabling precision agriculture practice. And next, uh, maybe that work can be done to explore and engineer beneficial plant microbe interactions to improve nutrient uptake, disease resistance, and overall plant growth. And next, maybe that developing plants with synthetic genetic circuits for improved photosynthesis, enhanced nutrient utilization, and resistance to pest and disease. Work can be also done to know how epigenetic modification contribute plant adaptation and stress response. Continuing to refine and advance gene editing tools such as CRISPR-Cas. Although CRISPR-Cas is very efficient tool, but still it need refinement. And another is investigating potential of plants for producing bioactive compounds with pharmaceutical application. This is another area of research. So overall conclusion is that future direction in plant biology involve a holistic approach integrating molecular, genetic, ecological, and technological perspectives. As the world faces challenges related to climate change, food security, and environmental sustainability, plant biology remains at the forefront of scientific innovation, offering solutions to ensure a resilient and nourished future. Thank you.
Thank you so much, sir, for your guidance.
Okay.
Thank you. Now, I would again request to Shinde ma'am to introduce our next guest, Dr. Wadikar sir. Shinde ma'am.
Now I am here to introduce our second resource person, Professor Dr. Wadikar sir. Dr. Wadikar sir is head of the Department of Botany at RB Attal College, Geurai. Sir worked in many colleges as associate professor and professor. In 2003, Sir completed PhD in studies on charcoal rot management of pigeon pea. He has a 21 years of research experience and also works as a research guide. Under his guidance, 6 students awarded PhD and 5 students are pursuing PhD. Sir completed one research project funded by UGC. He has published 2 textbooks, 61 research papers, and 4 articles in various journals. Sir worked as convener of state level and national level conference. He is a member of many educational committees and many journals. Thank you for this opportunity to introduce our guest, respected Wadikar. Thank you.
Thank you, ma'am. Now I would like to call or invite Honorable Wadikar sir to share his views on recent trends in plant science. Wadikar sir. Wadikar sir.
Hello, sir. Hello.
हाँ वाडीकर सर.
Can you audible, sir, me?
Yes sir, yes.
Good afternoon, respected chairman of today's national seminar on recent trend in plant sciences, Dr. Professor Shivdas Sirsat, sir, Principal Yashwantrao Chauhan College, Ambejogai. My dear friend, Dr. Deepak Bharti, Professor and Head Economics. My friend, Dr. Arvind Godke, sir, Vice Principal, Dr. Ramesh Shinde, sir, Dr. A.M. Barure, madam, Convener Gayatri, madam, and all respected participants. My topic is studies on bioactive compounds from citrus fruit in disease resistance. We believe that major three challenges for humankind in the 21st century are food, energy, and the environment, including climate change and environmental degradation due to pollution and habitat loss. In a word, sustainability, plant life play as an essential role in all three of the sustainability challenges. All of our food and the majority of our energy are produced by photosynthetic plant. Plants are major players in determining our climate, and agriculture expansion is a major factor in habitat encroachment and pollution of waterways by fertilizer application and runoff. Furthermore, these issues are not independent as the climate change. Additional challenges are placed on plant performance and thus food supply and habitat. Research on plant is needed to provide solution to these major challenges. Natural products such as plant extract, either as a pure compound or as a standardized extract, provided ultimate opportunities for new drug discoveries because of the unmatched availability of chemical diversity. According to World Health Organization, WHO, more than 80% of the world population relies on traditional medicine for their primary healthcare needs. The use of herbal medicine in Asia represent a long history of human interaction with the environment. Plants used for traditional medicine contain a wide range of substances that can be used to treat chronic as well as infectious diseases. Modern lifestyle is associated with several health issues. Humans are daily confronted with the presence of various environmental pollutants, including allergens, microbes, and the various other contaminants. The above and other unhealthy habitat, such as smoking and ultraviolet radiation, are associated with high level of free radical. Their excessive production causes damage to various cellular structure and consequently leads to chronic disorder such as cardiovascular diseases, rheumatoids, arthritis, respiratory problem, diabetes, and cancer. Antioxidants play an important role in maintaining balanced cellular function by neutralizing free radicals through various mechanisms. Food industries widely use synthetic antioxidants to extend shelf life and improve the quality and safety of their food products. However, such synthetic antioxidants are not preferred due to toxicity concern. Therefore, consumer interests have focused on identifying natural extract that can be used as a natural antioxidant since they are considered safer. In recent years, the interest in natural extract from medicinal plant has grown exponentially due to the rich content of the phytochemical substance like bioactive compounds and bioactive compounds from citrus fruit in disease resistance. Introduction. Citrus is genus of flowering plant in the family Rutaceae around that is commonly known as orange family. मराठीमध्ये आपण लिंबूची फॅमिली असे म्हणतो. Citrus fruit are grown mainly in tropical and subtropical countries of the world such as India, China, USA, West India, Malaya, Mexico, Australia, Brazil, Spain, and Italy. In India, they are grown in mainly Maharashtra, Andhra Pradesh, Punjab, Karnataka, Uttar Pradesh, and Bihar. The most important commercially citrus cultivars in India are the orange, followed by sweet orange and lemon. In Maharashtra, the citrus fruit are grown mainly in Chhatrapati Sambhajinagar, Jalna, Ahmednagar, Pune, Nagpur, and Solapur district. Citrus fruit are delicious, refreshing to eat, provide vitamins, minerals, and are available throughout the year. citrus peel are primarily byproducts. During processing, discarded as a waste and present an environmental problem. Peel has biologically active compounds, including natural antioxidant, and can be used as a cheap source of functional ingredient and food additives. The rind of citrus fruit is rich in a pectin, certain essential oils, and glycoside. Therefore, most of the researchers have claimed that the fruit, which are rich in antimicrobial chemicals in the rind at any stage of the fruit, provide to resistance for the disease caused by microorganism. The high concentration of various sugars, minerals, and amino acid also provide a good platform for the successful and survival of various microorganisms. Significance of the work. Peel of various fruits are generally considered as waste products and are normally thrown away by us. From different studies concluded a peel revealed that presence of important constituent. Also, it has been found that peel of fruit hold a tremendous potential to serve as a source of newer, effective, safer, and better antimicrobial agent. There are always an increased attention in bringing useful product from waste material, and citrus waste are no exception. Suitable method have to be adopted to utilize for the conversion into value-added products. Byproducts, recovery from the fruit waste can improve their overall economic processing unit. Besides this, the problem of environmental pollution also can be reduced considerably. The citrus peels are rich in nutrient and contain many phytochemicals. They can be efficiently used as a drug or as a food supplement too. Peel represent between 50 to 65% of total weight of the fruits and remain as the primary byproducts. If not considered further, it became waste produce, odor, soil pollution, herborage for insect, and can give rise to serious environmental pollution. It is thus significant and even essential to find the potent application for these peels. The present study was undertaken with the following objective, that is a collection of citrus fruit from different regions of Marathwada, studies on post-harvested diseases of citrus fruit, isolation and identification of pathogen from post-harvest citrus fruit like orange, Citrus reticulata, Citrus sinensis, Citrus aurantium, and Citrus aurantifolia. To study effect of carbon, nitrogen, amino acids, chemical like fungicides and organic acid. To study effect of different nutrients on enzyme activity and toxicity. Eco-friendly management of post-harvest fungi. To study the efficacy of fruit rind extract. To study and find out the most significant fruit with highest antimicrobial activity, antioxidant activity, and phytochemical analysis of citrus fruit rind. Collection of samples. Citrus fruit samples were collected from the different localities of Marathwada region in Maharashtra, especially Silod, Bid, Jalna. Studies on post-harvest diseases in citrus fruit. Symptomology were studied on different citrus fruit diseases. Isolation of fungi. Fungi were isolated on fungal media like PDA, that is the potato dextrose agar, identification of fungi. How to identify these various methods to use for the identification of the fungus, that is a morphology, colony color, margin of the colony, growth pattern and sporulation. The identification was made with the help of the manuals written by the different author, and the pathogenetic test with the help of the coach spatulate on the potato dextrose medium. Culture study effect of carbon for the growth of microorganism या any living organism. Carbon, nitrogen, amino acids are required in abundant quantity. That is a specifically effect of carbon, that is especially glucose peptone medium. Nitrogen sources, glucose peptone medium. Effect of amino acid, agar media. Effect of chemical fungicides on a CZA. Production of mycotoxin means any fungus या any microorganism secrete certain poisonous substances. That poisonous substances are known as a mycotoxin. Assay methods for the mycotoxin, that is a seed germination method. Effect of plant extract, that is a various plant product which can be obtained from the various part of the plant, that is a essential oil, fruit juice, fruit rind, plant gum, and antagonist activities, that is dual culture. Symptomology, that is means how to symptoms appears on the host by the pathogen, specifically Aspergillus. Aspergillus is one of the near about 2000 species of this fungus observed in different part of the world. Aspergillus niger, there is appearance of the fungal growth on the surface of the fruit and also from inside. At the latter stage, black moldy growth of Aspergillus niger appears on the whole fruit, and complete fruit became infected with the conidia emerging out. Alternaria citri, this fungus typically enters the fruit throughout the bottom and stem, and decay occur. Lesions developing from infection of the button became light brown to black in color and gradually progress over the fruit surface from the blackened button towards the styler end. Internally, the tissue turns black in the center of the fruit. Penicillium italicum. Penicillium is one of the fungus, and from that fungus wonder drug discovered by Alexander Fleming in 1942, and he awarded with the Nobel Prize with the discovery of the penicillin. But number of the penicillium species causes the number of diseases in number of important plant. Penicillium italicum, commonly known as a blue mold, caused by fungus Penicillium indicum, is another common post-harvest disease of lemon, but is usually less prevalent than green mold. Diseased tissue became soft, watery, and slightly discolored, and is easily punctured. The lesions enlarge more slowly those green mold. Aspergillus flavus, water soaked large spot affecting half or entire fruit and show fungus growth on the fruit and show dull green appearance of the fungus. Rhizoctonia bataticola, it causes infection on citrus fruit after harvesting when the fruit come in contact with the soil particle. Brown rot with grayish colored bands and sharply defined margin in areas where fruit appears, and the affected fruit later on became mushy. Phytophthora species, when fruit is infected by Phytophthora, is produced a decay in which the affected part became lighter brown in appearance, leathery and not sunken compared to the adjacent rind. Under humid condition, white mycelium grows on the rind surface. Diplodianthensis species, this disease remains dormant while the fruit is still attached to the tree. Once the fruit is removed and put into storage, symptom will be produced. The rind will first turn brown near the stem end of the fruit and then will progress down the fruit, forming brown finger-like streak. The whole fruit will eventually turn black and give off a sour fermented odor. Colatotrichum gliosporidaceae, anthracnose is caused by this Colatotrichum gliosporides and is also more common after prolonged period of rain. Fusarium oxysporium causes the fusarium rot of citrus fruit, but occasionally causes losses of citrus in the storage. Infected tissue is leathery, being to light or dark brown and sunken. Under humid condition, a white surface mycelium appears. These are the photoplate symptomology, that is a causative fungal pathogen on citrus fruit, that is a fungus Penicillium digitatum, Aspergillus niger, Rhizopus oryzae, Botrytis cinerea. Plate second, that is shows by the Botrytis cinerea, Penicillium italicum, Colatotrichum gliosporides, Alternaria citri. Plate next, that is a Penicillium digitatum, Phytophthora. Such spot which can be appear on the bread surface या chapati या bhakri surface. Isolation of fungi from seed orange, that is a fruit collected from different localities of Marathwada. The number of signs, that is a plus signs 3, which showing the high occurrence of the fungus, which is especially on that fruit which is located from that place. The colony plate and micro photograph of fungal pathogen on citrus. Suppose we can collect the citrus fruit from the market, and on that citrus fruit the fungus growth and that culture bring in the laboratory with the specific media. That name of that media is Zepextoc agar media, and the growth of the fungus which can be seen under the microscope, that is their fruiting body, that is a mycelium, and that is a colony of that fungus. This is आपल्याकडे मोठ्या प्रमाणात कांद्यावरती किंवा चिंचेवरती मोठ्या प्रमाणामध्ये आढळून येतो. The colony plate and the micro photograph of fungal pathogen on citrus fruit. This is a pure culture in a petri plate, which is showing these layers, that is a specific character of this fungus. That and these are the Fusarium oxysporium. आपल्याकडे तूर उधळते, त्या तूर उधळण्यासाठी कारणीभूत असण्यासाठी हा फंगस देखील कार्य करतो. The colony plate and micro photograph of fungal pathogen on citrus. These are the colony characters. Colony plate and micro photograph. Studies on fruit rotting at different stages in a orange. That is a unripe stage, moderately ripe, and overripe, means on which stage the fungal act more, means after the post harvest condition, the more appearance of the fungus occur on the fruit. Effect of carbon sources on the growth of the post-harvest fungi isolated from the citrus fruit. Carbon sources, that is the sucrose, starch, maltose, lactose, CMC, glucose. These are the carbohydrates and which having the large amount of the carbon, and on that content the fungus Rhizoctonia bataticola, Aspergillus niger, Penicillium digitatum and Fusarium oxyscorium, showing their growth on a different media. Effect of nitrogen. Nitrogen is required for the body of any organism because from the nitrogen protein synthesis is prominently takes place in body of the organism. That may be human being, that may be plant, and that may be microorganism. Effect of amino acids. Amino acids are required for the growth. 20 amino acids are required: leucine, phenylalanine, tryptophan, serine, proline, histidine, and these amino acids showing the activity on the specific Aspergillus niger, Penicillium digitatum, and Rhizoctonia bataticola. And this is a control. See the control value and the value of the amino acids in a particular days, that is 8 days, 10 days, 8 days, 6 days, 10 days. Effect of organic acids, various acids, that is stearic acid, acetic acid, malic acid, citric acid, tartaric acid, succinic acid, oxalic acid. These acids used for their growth of the Penicillium digitatum, Macropomena fazulina, Phytophthora, Rhizoctonia bataticola, and these are the values. Effect of plant extract on the for eco-friendly approach purpose means there is no harm to the nature as well as other microorganism. We used number of the plant, that is the Justica vesica, Aegle marmolas, that is commonly known as a bale, Dhatura metal, Dhotra, Vitex nigando, Nirgudi, Calotropis procera, Rui, Azadiracta indica, Neem, Oceum scanthum, Tudas, Ricinus communis, errant, Togatus platula, Trigus picumbanus. These are the different plants used for the eco-friendly approach, means to avoid the chemical pollution. Therefore, we can use this plant product for the minimization of the fungus. Any chemical do not control any pathogen because in nature the population of microorganism from 0 to 100, but never 0 and never 100. In between the range is a variable. Effect of essential oil, different oils, that is a neem oil, castor oil, almond oil, groundnut oil, clove oil, cinnamon oil, and these oils showing the effect on the different fungus, that is a Phytophthora, Penicillium digitatum, Rhizoctonia bataticola, whatever meat. Antimicrobial activity of citrus peel extract against gram-positive and gram-negative bacteria. Bacteria Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus pneumonia. These bacteria causes deadly diseases in human beings, especially infant babies, and on that controlling minimize their growth, that is different peel of the citrus species, that is a lemon, orange, sour orange, sweet orange, means they inhibit the growth of this particular bacteria. Antioxidant sample, that is activity sour orange, sweet orange, here 75%, here 83.40%. Qualitative phytochemical analysis of citrus fruit. Our research were chemically related with the phytochemical activity because phytochemicals which are specially bioactive compounds, that is specially terpenoids, saponin, flavonoids, tannin, phenols, and proteins. These are the antioxidant, and these antioxidant used for the various researcher for the chemical value as well as the herbal value to avoid the side effect of the medicine. Quantitative phytochemical analysis of citrus fruit, total alkaloid, total flavonoid, total phenolic content, tannin content, terpenoid, fixed oils, and fat. Phytochemical activity shows by this graph. Antagonistic activity. Antagonism means to control, to inhibit the growth of one organism with the help of the another organism. That process is known as antagonism. Trichoderma is one of the fungus which control minimize the growth of the Fusarium oxysporium. This is shown by this plate. Aspergillus niger, Penicillium digitatum, Fusarium oxysporium, Trichoderma viridi, Rhizoctonia bataticola, mucor species, and control. And these plants showing the toxicity by shoot cut method. These are the various terpenoids, saponin, flavonoids, tannin, phenols, and protein. Summary and conclusion. In survey, it was found that in Marathwada region, post-harvest diseases of citrus fruit were caused by different fungi like Aspergillus niger, Penicillium digitatum, Fusarium oxysporium, Colatotrichum gliosporides, Macrophomina, Rhizoctonia bataticola, Rhizopus stoloniferus, Mucor, Alternaria citri, Corularia, Aspergillus flavus, Phytophthora, Phomopsis citri, etc. Among these are Aspergillus niger and Penicillium digitatum species are common in all localities. It is clear from the observation that fruit ripening goes on increasing, the disease severity also goes on increasing. Different type of plant leaf extract were tested against pathogenic fungi. Among leaf extract of Azadirachta indica show significant reduction of radial growth of all tested fungi. Different types of essential oils were tested against pathogenic fungi. The inhibitory effect of the essential oils on pathogenic fungi shows that castor oil completely inhibited the radial growth of untested fungi. The screening of gum of selected plant against post-harvest citrus fungi in which Azadirachta indica show the antifungal activity against all tested fungi. Trichoderma viridi shows maximum antagonistic activity against Fusarium oxysporum. The most significant fruit with highest antimicrobial activity is orange, that is the Citrus reticulata. The result revealed that all extract of the orange peel shows highest inhibitory activity against all tested microorganisms like fungi as well as bacteria. To find out the toxic effect of fungi, that is Aspergillus niger, Penicillium digitatum, Fusarium oxysporum, Trichoderma viridi, Rhizoctonia bataticola, and mucor species were cultured and filtered, tested by shoot method, and it was found that the toxin production was noticed in Fusarium oxysporum, Trichoderma viridi, Rhizoctonia bataticola fungi, and among all the tested fungi, the cultural filtrate of fungus Trichoderma viridi shows maximum toxicity. Antioxidants were extracted from the fruit peels of orange, lemon, sweet orange, and sour orange. Maximum antioxidant activity was found in orange, followed by sweet orange, lemon, and minimum in sour orange peel. The preliminary qualitative phytochemicals investigation documented that the peel extract of all 4 citrus fruit show the presence of many bioactive compounds, namely phenols, flavonoids, terpenoids, saponin, alkaloids, and tannin. The result revealed that in orange and sweet orange peel extract, all phytochemical listed above are present. In lemon peel extract, only flavonoid absent and other phytochemicals are present. In sour orange peel extract, flavonoid present and other phytochemicals absent. The qualitative phytochemicals investigation documented in the orange peel extract phenolic content, alkaloid, flavonoid, terpenoid present high percentage than sweet orange peel extract, but tannin and fixed oil and fat are present in high amount in sweet orange peel extract. Depending upon the result procured from the investigation, it can be concluded that peel of orange, that is the Citrus reticulata, sweet Citrus sinensis, lemon Citrus aurantium, and sour orange Citrus aurantifolia exhibit antimicrobial activities. This work has highlighted the antimicrobial effect of citrus plant extract. This investigation opens a new way and suggests the efficacy of this extract as antimicrobial factor. The fruit rind may be used as a guiding path in continuing search for new natural product with potential medicine attribute, as it will lead to new development of bioactive compound to act as antimicrobial agent. The work carried out not only might help us providing insight into better uses of fruit waste and help reduction of solid waste, but also help us in understanding the nature of compound and their level in their fruit waste. From the above applied workout, it can be clearly concluded that citrus fruit having high phenolic, alkaloid, flavonoid, terpenoid content, future research will be helpful in the isolation and separation of this compound content so that it could be used for human welfare. Thank you. I am very much thankful to Principal Dr. Shivadas sir and convener Gayatri Kadam madam and my friend Arvind Ghodke sir. Thank you, sir.
Thank you, sir. Thank you for your speech. It will be definitely helpful for us in future. Thank you. Now I would like to call Power Ma'am for vote of thanks. Power Ma'am.
Thank you, madam. Good afternoon to all. I am Sneha Pawar. Feel very grateful to present here vote of thanks on this occasion. On behalf of Yeshwantrao Chauhan College, Ambazogai, I would like to thank keynote speaker and resource person, Honorable Dr. Mehta sir. We really enlightened with your guidance and presence. I would like to thank our resource person, Honorable Dr. Wadikar sir, for spending precious time with us and guiding us on a topic studies on bioactive compound from citrus fruits in a disease resistance. I would like to thank President of today's seminar, Honorable Dr. Sisat sir and White Principal, Dr. Shinde sir and Zado sir for motivation and support. Thank you, Dr. Ghodke sir, our science coordinator who always support us to work hard and do our best. Thank you, Kadam madam, for organizing today's seminar, and thank you, Shinde madam, for introducing our guest. I also want to thank all of you for joining and helping us to make this day successful and special for all of us. Thank you. Here, with President permission, I announce that the program is concluded. Thank you so much.